Hello, my name is Tamer Whittle-Haig, and I will today be presenting on our research study which looks at the mechanisms underlying microbial-mediated changes in social behavior in mice models of autism spectrum disorder, or ASD for short. Alright, so let's get into what is autism spectrum disorder. There are a lot of behaviors associated with autism spectrum disorder in kids which are used to diagnose it, some of which, according to the CDC, include not looking at objects when another person points at them, having trouble relating to others or not having an interest in other people at all, avoiding contact and wanting to be alone, etc. Autism exists on a huge spectrum, meaning that it can be quite obvious to tell when somebody has a disability, or you can be completely blind to it. There has been cases where people with autism spectrum disorder have been associated with being extremely gifted in usually in one particular subject as well. All right, this is where our study comes in. We are hypothesizing that L. retori, a bacteria found in the microbiome, can treat some of the social symptoms of autism. So we hypothesize that L. retori can reduce social deficits in ASD mice models by acting in a mechanism involving the vagus nerve. And a bit of background on the vagus nerve: it's a nerve and one of the many connections between the gut and the brain. Additionally, we hypothesize that L. retori can affect the oxytocin dopamine reward system, and the oxytocin dopamine reward system can also play a role in ASD, as it has been previously found that oxytocin can improve social functioning in ASD subjects. All right, on to our methods. So, the different mice models included genetic, environmental, and idiopathic mice models of ASD. Rats were either Shank3B, WT, meaning it's a control, or Shank3B plus vehicle, meaning they had ASD characteristics and were not getting treatment. Mice were found to be models of ASD based on a number of behavioral/molecular tests. For example, mice which lack the Shank-B isoform of the Shank3 gene are known to inhibit ASD-like behaviors. Treatment of L. retori was added to daily drinking water for experimental groups, while control groups received a consistent volume of water without the bacterial treatment. After this, a lot of bacterial samples were collected. There was behavioral assays, tissue collection, and electrophysiological recordings, but I won't go into the specifics of each test. On to our results. The first result included testing the mice named Shank3B-, which lacked the Shank3B isoform of the Shank3 gene. And as a reminder, the Shank3 gene, mice which lack the isoform exhibit ASD-like behaviors. In these specific mice, we wanted to see the alteration of the Shank3B mice microbiome and the prevalence of L. retori bacteria. This was performed via 16S rRNA gene sequencing, and it was found that L. retori levels were significantly lower in the Shank3B- mice than the control mice. As you can see here on the slide, the red is the Shank3B, and the L. retori levels are a lot lower than the control, the WT, and there's significant difference between the two groups. Alright, on to result 2. After treating Shank3B- mice with L. retori, there is an overall increase in sociability. The behavior of the Shank3B- mice even mimicked the behavior of the control mice. Furthermore, L. retori was not just found to reverse the ASD sociability in one particular ASD animal model, but all 3 ASD animal models: genetic, environmental, and idiopathic. The mechanism by which L. retori was found to improve social behavior was that L. retori acts on the vagus nerve, and essentially this means that L. retori is dependent on the vagus nerve being intact and working. Without the vagus nerve, supplementation of L. retori does not increase sociability in Shank3B- mice. So in this result, you can see that the blue dots are vagotomized Shank3B-, and vagotomized mean that they didn't have their vagus nerve intact and working properly. And you can see that those who had the vagus nerve working properly had higher sociability, scored higher in the sociability tests. So you can see that for the interaction times. Alright, moving on to result 3. It was excitingly found that L-retorrhea increases oxytocin levels by increasing both the number and fluorescence intensity of oxytocin-positive neurons in Shank3B- mice. In this graph, you can see how both the control and the Shank3B- mice treated with L-retorrhea show a similar level and intensity of oxytocin-positive neurons, while the Shank3B plus vehicle, the sham, has reduced levels and intensity of oxytocin-positive neurons. It is known that an increase in oxytocin can increase dopamine levels in the VTA, a part of the brain partially responsible for social interaction. So what this means is that can better be described by result 4. So here in result 4, it's more about the VTA. It was also found that social interaction induced VTA plasticity of the Shank3B vehicle is less than that in mice with ASD-like traits. And this means that they have a quote-unquote weak VTA, as theoretically it is not able to change, adapt, or perhaps mature with development. Conversely, L-retorrhea treated ASD mice improved the social interaction-induced synaptic plasticity in the VTA. So if you remember back to our last slide, how you saw that there was a decrease in the levels of oxytocin-positive neurons in the Shank3B- and Shank3B plus vehicle, that it kind of means that these intensities, the density of these neurons, they're affecting the plasticity in the VTA. And then this result, it's showing that what that is doing is affecting the ratio of AMPA to NMD receptors in the VTA. And you can see here on the graphs how in the different in the controls, the Shank3B minus, the Shank3B plus vehicle plus L-retorrhea, whatnot, when they were treated with L-retorrhea, there was an increase in the AMPA to NMDA ratio, which is a good thing for the VTA plasticity. Alright, so moving on to our discussion. Because the Shank3B- mice displayed an increase in sociability compared to the control mice when given L-retorrhea, this seems to further indicate that species richness plays an important role in our brain. For example, since pre- and postnatal periods are critical developmental windows that influence lifetime behavior, it is important that the microbiome is as developed and as healthy as possible during these periods. So let's talk about what may change the microbiome during these critical periods of development. One of the correlations mentioned earlier was how it was found that an increase, that that you're more likely to have autism if your mother takes a certain medication like aproic acid while pregnant. It could be possible that these medications could have an effect directly on the baby's microbiome or the mother's biome, which could in turn affect the baby. The finding that L-retorrhea increases oxytocin levels, leading to an improvement sociability in ASD animals, means that it is likely that some of the ways in which the gut can influence the brain is through the quantity of neurotransmitters. The known mechanism by which it is thought that L-retorrhea plays a role is via the vagal nerve fibers projecting to the PVN, where oxytocin is produced. It is thought that L-retorrhea increases vagal nerve activity, which in turn increases the activity of the PVN. With the increases in oxytocin produced by the PVN, it is thought that this goes on to activate neurons in the VTA, which again is a region in the brain partially responsible for social behaviors. The finding that L-retorrhea treated ASD mice improved the social interaction induced synaptic plasticity in the VTA suggests that the decreased sociability trait that comes with autism has a real hope of being reversed. If the VTA can respond to microbial treatment and improve its plasticity, then it is likely that other areas of the brain impacted by species richness from the microbiome can be improved as well. Overall, the paper found that L-retorrhea is important for social behavior, and in future research, we think that it is important to explore how some of the other aspects of certain bacteria can influence other areas of the brain, looking at L-retorrhea richness in other mammals and their social behavior, and lastly learning if L-retorrhea only works through the vagal nerve or if it influences the brain in another way. To wrap it up, our study did an amazing job with the number of trials ran and the number of mice used, but it was worth ourselves to ask why we chose to feed the Shank 3B- with Elratory for a period of 4 weeks and not longer or shorter. Thank you.